Good morning, students. I am Shomajit sir. Today I am here to deliver my lecture on chapter Biomolecules. So today we'll start with a fresh new chapter, Biomolecules. Here I'll discuss about the introduction of biomolecules and also I'll discuss about amino acid. So without any delay, let's start with the chapter Biomolecules. Here I'll discuss about the introduction of biomolecules and amino acid. So let's start. So let's start with chapter Biomolecules. You can see very interesting diagram I have given. It's a structure of DNA, the double helical structure, and right side you can see a lot of proteins are there. Now biomolecules, all right. So biomolecules are chemical compound found in living organisms. So in the living organism, biomolecules are found, and they also include organic and inorganic compound. All right. So biomolecules are living compound that are found in a living organism, and these include or they include both organic and inorganic compounds. Now, how we can analyze the chemical composition in a tissue? That means in a tissue, what are the chemical compounds are there? We'll do one experiment. Here first we'll grind a living tissue. It can be a vegetable or a piece of liver, etc. And now what we do, in that grinded tissue we add it trichloroacetic acid. We add trichloroacetic acid to get a thick slurry. All right. Now we'll strain this through a cheesecloth or a cotton to get two fractions such as filtrate and the retention. The filtrate part will be the acid soluble pool and the retention part will be the acid insoluble fractions. Alright. Now we'll discuss what is a filtrate and what is a retention part. So in the filtrate part, that is the acid soluble pool, it contains biomolecules or biomolecule weight less than 1000 Dalton, whereas in the retention part, that means acid insoluble fraction, it contains biomacromolecules, that means having a molecular weight that is greater than 1000 Dalton. So what we find here, in acid soluble pool we find biomolecules, and in acid insoluble fractions we find biomacromolecules. So here in this slide we have understood that in the acid soluble pool or in the filtrate part, biomolecules are present that is having a molecular weight less than 1000 Dalton, whereas the retention part or acid insoluble fraction, biomacromolecules are found that is having a molecular weight greater than 1000 Dalton. Now let's proceed with the next slide. Next slide is analysis of inorganic compound. How we can analyze an inorganic compound? First we'll weight a living tissue, that means we'll take a weight and it is dried, and we'll take dry weight to dry to evaporate water. So we'll take a weight and a dry weight. So it is fully burnt to oxidize all carbon compounds to gaseous form, that means carbon dioxide and water vapor, and it forms ash. So the ash contains inorganic elements. All right. So first what we did, we took a living tissue, we weighted, and then we dried it to evaporate water, and then again we took a dry wet, and it was fully burnt, all right, to oxidize all carbon compound in a gaseous form, so that when it was burnt, the carbon dioxide and water vapor were lost and it forms ash. And this ash contain inorganic elements, that is calcium, magnesium, sodium and potassium, and inorganic compounds like sulfate, phosphate, sodium chloride and calcium carbonates were there. So you have to understand, or you have to memorize the inorganic elements and the inorganic compounds. So inorganic elements are calcium, magnesium, sodium and potassium, and inorganic compounds are sulfate, phosphate, sodium chloride, calcium carbonate, etc. So next we see the comparison of elements in living and non-living matter. All right, so what are the different comparison of living matter and non-living matter? We have in the left hand side the elements, and then in the right hand side the percentage of weight. All right, in the earth crust and the human body, we see the elements are hydrogen, carbon, oxygen, nitrogen, sulfur, sodium, calcium, magnesium, and silicon. All the symbols are given, and you all know hydrogen symbol is H. And you can see the different comparisons are given, like hydrogen in the earth crust is found 0.14, whereas in human body it is found 0.5. All right, like silicon in the earth crust is found 27.7, whereas in the human body is negligible. Like in sulfur in the earth crust is found 0.03, whereas in the human body is found only 0.3. So we move to the next slide. We'll start with bio micromolecules. Now, what are micromolecules? Now, what are biomicromolecules? Just now we have explained that biomicromolecules is micro means small. All right. So, molecular weight of biomicromolecules, that means micromolecules or biomolecules found in an acid soluble pool, ranges from 18 to 800 Dalton. The short form of symbol of Dalton is Da, and the acid soluble pool represent the cytoplasmic composition. All right, so biomicromolecules include amino acid, lipids, sugar, and nitrogen base. So what are the different components of biomolecules or biomicromolecules? Contains what? Amino acid, lipids, sugar, and nitrogen base. Here in this video lecture we'll discuss only about amino acids. All right, so one thing you have to understand, the molecular weight ranging from 18 to 800 Dalton. All right, those are called biomicromolecules. So in the biomicromolecules portion, we'll start first with amino acid. All right, so amino acid is the unit of protein. So amino acid is an unit of protein. You can see in the right hand side the structure of amino acid is given. You can see, we know that carbon has 4 hands, isn't it? So here carbon is there and 4 radiating hands are there. So in one hand, in the right hand is there carboxylic group, that is COOH group, and the left hand side is amino groups NH. Right, and in the third hand is the hydrogen, and fourth hand, that is R group, alkyl group. It's a variable side chains. All right, so they are the compounds formed of amino group, that is NH2, and acidic group, that is a carboxylic group, that is COH, and H and variable R group. All right, so amino group will be NH2 and carboxylic group will be COH. So NH2 and COH are attached to same carbon compound, that means always in the alpha chain, straight line it has to be attached. So they are called alpha amino acid, and they are substituted methanes. Now you can see in this amino acid chain, we know that carbon has a four hands. One hand there will be a carboxylic, another one there will be amino group. All right, so wherever in another hand there will be H, and in fourth hand there will be R means alkyl group. Wherever R, instead of R there is H, that amino acid is called glycine. Wherever in place of R there is methyl, that is CH3 group, it's called alanine. Amino acid alanine, and short form of alanine is ALA, and short form of glycine is GLY. And wherever in place of R there is CH2OH added, it's called serine amino acid. All right, so you can see 3 amino acid structures are given. First one is glycine, then alanine, and then serine. Now there are 20 types of amino acid that are used in protein synthesis, and these amino acids are of following types. We will see, one is acidic amino acid, basic amino acid, and neutral amino acid. Acidic amino acid means it will contain carboxylic group. All right, 2 carboxylic group will be there. Basic amino acid, at least 2 amino group it should have, and neutral amino acid group, 1 carboxylic and 1 amine will be there. All right, So you can see examples are there in acidic amino acid, that is glutamic acid, basic amino acid, that is lysine, and neutral amino acid is valine. So some amino acids are aromatic, example tyrosine, phenylalanine, tryptophan. All right. So in the previous structures of amino acid, I have said that in the carbon has four rotating arm, one will be carboxyl, another will be amino group. All right. So in a amino acid having two carboxylic acid and one amino group, that is acidic. So in a amino acid having two amino group and one carboxylic, that is basic. And in a neutral, there is a one carboxylic and one amino group. So more the carboxylic group in an amino acid is called acidic, more the base in an amino acid, the basic. That more the amino group NH2 called basic, and neutral is one will be carboxylic, another will be amino group. Now we'll talk about the amino acid can be classified as follows. Two different types of amino acids are there. One are essential amino acid, another are non-essential amino acid. So non-essential amino acid and essential amino acid, so they cannot be synthesized by the body. So essential amino acid cannot be synthesized by our body and should be supplied through diet. All right. So essential amino acid are not synthesized by our body and are supplied through diet, whereas non-essential amino acid, they can be synthesized by the body. So examples are lysine, leucine, isoleucine, and tryptophan. Whereas in non-essential amino acid, the examples are glycine, alanine, serine, and arginine. All right. So essential means those are not synthesized in our body. We have to take through diet, and non-essential means they are synthesized in our body. So next you see in an amino acid, the amino group and carboxyl group have ionizable nature. So the structure of amino acid changes in solutions of different pH. So if both amino group and carboxylic groups are ionized, then it is called zwitter ion. All right. So that means amino group NH2 will be ionized into NH3 plus. All right, that means NH3 it will be ionized into. So we can 3 there, one H3 plus N, that is ionized form of amino group. And you can see the ionized form of carboxylic group is COO minus. All right, so COO minus, and this side N plus H3 or H3 plus N is a positive side, that's deuterion form. All right. Thank you, students. This was all about the entire video lecture. We have discussed about the introduction of biomolecules and amino acid. So stay at home, stay safe. I am Shomuji sir signing off for today, and again we'll meet in the next class. Thank you, students.